This session is intended for our post-laboratory discussion or post-laboratory notes for module number 6, the demonstration of the nitrogen cycle as our model biogeochemical cycle, where microbes, the activities of microbes are heavily involved for the process to be sustainable or for the process to continue. So, we've discussed a lot about the nitrogen cycle. So I won't be providing any additional information anymore. But for this activity, the focus that we or our main achievement is um, we're able to understand four key processes in the nitrogen cycle that involves activities of microbes. It involves microbes and their associated physiological activities. So, those four events or four processes in the nitrogen cycle that we demonstrated using our with the protocol that we have include ammonification, no? Then we also have nitrification, both for nitrite and nitrate. And we also have denitrification. And then somehow we also introduce the idea of nitrogen fixation. No? So we begin with ammonification. So we've defined ammonification as one of the key events, no? One of the initial events in the cycle of elemental nitrogen. So in ammonification, we expect that there is conversion of organic carbon, the reduced organic carbon, to a form that is inorganic. No? So you produce your nitrogen atom associated with non-carbon atoms. So yung NH3 and NH4. No? And the process, ammonification, is ano no microblend. So it's primarily facilitated by different types of microbes. So in our demonstration, in our experiment, no, we used peptone broth as our substrate. So that's the source of our nitrogen, of our organic nitrogen. And then we used different setups. We have a setup containing soil, a setup containing Bacillus cereus, a setup containing Pseudomonas fluorescens, and another setup that use Proteus vulgaris. So for this part of the module, part of the experiment, we tested for the presence of ammonia using a reagent called the Nestler's reagent. No? And then we also determined the milligrams or the amount of ammonia that was produced per 100 ml. No? Hypothetically, now given a set of variables, we can compute no, for the amount of ammonia present in the solution that we tested. Okay, so let's take a look at the worksheet data. No? So, medyo na break ko na yung routine natin where you're supposed to submit the worksheet before I provide you the answers or the post lab. Anyway, for the purpose of being able to review properly, no, I'm giving this worksheet away. But for the for the guide questions, I won't be answering the guide questions anymore. I'll just discuss the hypothetical data and then some narratives we have to remember. Let's take a look at the first table, no, sa ammonification. Yan. Okay, so this equation here or this reaction here is called the Nestlerization reaction. No, that a molecule of ammonia once coexists with the Nestler's reagent, or once you add the Nestler's reagent, no, it will react with it, forming a yellow brownish colored chemical species, no, and that's what we call mercuric ammonia. No? So in the ammonia, in the event that ammonia is present in your setup with Nestler's reagent, the colorimetric indication is dapat may yellow or brown na ano no colored chemical species. So that's mercuric ammonia. Okay, now let's take a look at the table of our different setups. So meron tayong four setups, and then we have of course our negative control. Okay, so we have soil, bacillus, pseudomonas, and proteus. Okay, as I've discussed and repeated multiple times already, expect that most terrestrial microbes will really have the capacity to break down organic compounds containing nitrogen so that they can produce inorganic nitrogen containing compounds like ammonium, ammonia. So most of microbes possess that, most terrestrial microbes. That's why it is expected that all the tested microbes here, they will be producing ammonia, and they will of course react with the Nestler's reagent. Ang difference na lang siguro is more on the intensity of the colored compound. Probably if we tested this face to face, most likely makikita nyo may mga differences na it could be that there is may ranking to, but of course we cannot verify that. We don't have any empirical data as of the moment. So let's just play safe and then assume na all of the terrestrial microbes can facilitate ammonification. Okay? So, but we expect, of course, yung soil sample natin will have the most, the highest intensity of the yellow colored chemical species production. Yan ang may pinakamataas kasi multiple species yan ng mga bacteria. So you expect combined rates of ammonification. Okay? Now, if we take a look at the succeeding steps, diba, you're asked to check on the amount of ammonia produced. Okay, so since we verified, we hypothetically lahat to dapat nag-produce ng ammonia. The next part is we titrate or we do titration in order to determine the amount of ammonia produced. No. Okay, so in order to do that, the protocol employed that we use H2SO4 0.1 N H2SO4. No, so as expected, no, the soil will of course have the greatest requirement for the acidic chemical to be added para matitrate siya para matitrate siya successfully. We expect that it will the soil setup will have the most requirement in terms of H2SO4, while the others will of course vary, and the control will require probably very one drop lang, no, and then okay na siya, titrated na siya kasi of course we expect sa control neutral na agad ang niyan ang pH niyan. No? Okay, now next column. Okay, the amount of NH3 that is produced signified or indicate quantified in terms of mg per 100 ml. So as expected, it's the soil setup that is expected na dapat siya yung may pinakamaraming ammonia produced, while the others vary na lang, and control kung meron man very low or totally absent siya. Now, let's take a look at how we compute for the milligrams of ammonia per 100 ml. So, in our protocol, we are asked to withdraw or we use 5 ml of the medium from each of the tubes containing these. And then we add 3 to 5 drops of the methyl red indicator to determine kung basic ba or acidic yung pH. But of course, we expect during ammonification, or during ammonia production, tataas ang pH, no? Magiging basic ang pH ng setup. So we have to titrate it. Kailangan gawin natin 7, no? Kailangan natin i-neutralize. Now, how do we compute for the amount of ammonia produced? So we just have to follow this formula. the formula is here, no? Milligrams of NH3 in 100 ml is equivalent to the value of T multiplied by the normality of the acid multiplied by the equivalent weight of NH3 multiplied by 100 divided by the volume of sample. Okay? Now, before we look at the formula, so some quantities muna we have to define. The value of T is the difference between the ml that you use in the sample, that's 5, and the ml that you use in the control, no? ml of acid that was used in your control, no? So, let's say for example na let's say, no, for example, in one of the setups, you use 5 ml of acid to neutralize your sample, and then 5 ml para naman sa control. So dapat magiging ano siya, no, magiging zero siya. So but for the purposes of, making sure that meron tayong number na ma-produce. Let's say for example, you used 5 ml of your titrating agent in your sample, and then 0.5 ml ang ginamit mo for your control, yung natin, negative control natin. The value of T will be equivalent to 4.5. Okay? So you have to just get the numerator, multiply lang lahat yung values na yan, and then you divide the volume, divide it by the volume of the sample that was tested. So for our case, it's 5 ml. So the computation will yield as follows. So 4.5 times 0.1 times 14 times 100 divided by 5, that's going to be 126 milligrams of ammonia per 100 ml. Okay? So that's, I'd say an equation that we can use if we want to sort of quantify yung ammonia production by a certain by a certain setup where meron kang bacteria capable of carrying out ammonification. Okay, but this is again a demonstration equation lang siya. It's not really something that is standard, but relative, or if we are to estimate, we can use this formula. Okay? So very straightforward yung first part ng ating experiment. The second part deals with the process of nitrification. So we've defined nitrification in the lecture. We discussed it as the biological process of ammonia oxidation. Diba? The product of nitrification is either nitrite or nitrate. Diba? So nitrite, actually these two outputs from ammonia biological oxidation of ammonia in nitrite and nitrate, they can be produced sequentially, or they can be produced or pwede rin naman na from ammonia diretso na to nitrate, no? And that process is actually carried out by a special group of bacteria called the COMAMOX bacteria, the complete ammonia oxidizing bacteria. So instead of producing nitrite with COMAMOX bacteria, we can expect the biological oxidation, a direct biological oxidation of ammonia to nitrate. So binabypass na yung nitrite na stage, no? Okay, so usually we expect that same with ammonification, of course, no, in the context of soil, terrestrial environments. This happens a lot in the soil, no? And a lot of bacteria are capable of carrying out nitrification. Different genera of bacteria are capable of carrying out nitrification. Okay, so here we have one setup. So meron tayong setup where we put soil, 0.5 grams of soil, and then we incubated it for 25 to 30 degrees Celsius. And then we use a special solution called the Tromsdorf solution in order to check the presence of nitrite, no? And then we test the culture for the presence of ammonia once a week. For nitrate formation naman, it's the same setup. We also use the Tromsdorf solution, but in the for the purpose of ensuring that nitrite is gone. No? So, wala nang nitrite can be indicated by the colorimetric reaction of the Tromsdorf reagent with the sample, no? The setup that we're testing. And then afterwards, we use the DPA test, the diphenylamine test, in order to determine kung meron namang nitrate. Okay? Let's take a look at the data, associated data na tayo here. Okay, so for nitrification, yan. Okay, so here's the expected data. For soil, we expect that the Tromsdorf's reaction under the nitrite part, we expect that of course, no, there is blue-black coloration. I'll explain later bakit siya blue-black. No? And for control, there's no pigmented chemical species that is expected to be produced. Now, testing the sample with for the presence of NH3, no? So we expect that as time passes by, 'di ba, si ammonia, mauubos yan, ma-oxidize siya totally to nitrite. So eventually there is from a positive detection to a negative detection or absence, total absence of NH3, no? And of course, nitrites will be present. For the control, of course, dapat negative yan kasi nga comparator natin siya. Okay? All right, so let's take a look at the process of ammonia oxidation. So ammonia oxidation yields nitrite and nitrate, no, sequentially. Though not all the time, you expect nitrite to be present, no? Because there, again, as I mentioned, there are some microbes capable of directly oxidizing ammonia to nitrate, no? In normal or in microenvironments, you don't expect these compounds, these molecules, to stay stable for a long time. So what do you call them? They convert, no? Ang turnover ng sinks ng nitrite and nitrate masyadong mabilis yan. Hindi sila they won't stay stable for a long time. Okay? So they will transform from one compound to another. Okay. Si nitrite, that's the one that's being detected by the Tromsdorf reagent. No, it will give a blue-black colored chemical species. No, nitrate will of course expected to be produced, and in the process of nitrate being reduced back to nitrite, no, it can just incorporate itself to nitrogen-containing gas, molecular dinitrogen or other gases with nitrogen. No? Now, the Tromsdorf reagent, no, how is the Tromsdorf reagent capable of detecting nitrite? Let's take a look first at the components of the reagent. So, according to the MSDS paper ng Tromsdorf reagent, yung MSDS, yung material safety documentation niya. Ito ang composition ng Tromsdorf reagent. So there are three chemical components: zinc chloride, potato starch, and potassium iodide. Okay? Now, initially, once you add your Tromsdorf reagent doon sa setup mo and you expect nitrites to be present on your setup, those nitrites on your setup will be reacting with the Tromsdorf reagent and it will produce nitrous acid. Okay? That nitrous acid can oxidize the iodide present into iodine. And we know that iodine is a big molecule, right? It's a big atom. It's a big atom, diba? And it can complex with different other atoms or compounds around it. That's normally nagpo-produce siya ng colored na complexes. So the iodine can form a blue-colored complex with starch, yung starch component. Kaya parang magkakaroon ka ng blue-black na coloration. And that indicates na indeed nitrite is present. Kasi without nitrite, no nitrous acid will be produced, no iodide will be produced. So very important, what do you call this, without nitrite, totally your sample hindi siya magre-react with the Tromsdorf reagent. Okay? Alright, so, ayan no? So na-mention ko na rin yung further oxidation ng nitrite into nitrate, NO3. So, nitrate, it can react with, it can react with zinc metal, with zinc, elemental zinc, no? And that zinc is going to be oxidized while nitrate will be reduced to nitrite again. So kung merong mga compound, may mga nitrates man dun sa sample, sabihin natin no, na napoproduce siya, okay, it may go back to being nitrites again, primarily because of the presence of the zinc atom from zinc chloride. Okay? Now, for nitrate detection, no, nitrate produced in the inoculated nitrate-forming medium. Okay, so here, I'm just going to make this one table. Yeah. Okay, so we have two setups, one soil and one control setup. Okay, so for soil, we expect no, that there's no pigmented chemical species produced. Primarily kasi yun nga part ng protocol natin na as we proceed with with nitrate formation, dapat walang nitrite na mapoproduce, no? So you expect na magne-negative talaga siya. And the color reaction with diphenylamine reagent, expected of course for the soil should be blue coloration, or it can be varied depending on the amount of the nitrate, no, that is present on the setup. And for control, of course, it's negative. And presence of nitrates, obviously positive siya dapat, no? Since you expect the blue coloration to be produced. Now, that blue coloration is actually attributed to the reaction between diphenylamine reagent and the nitrate ions. So they will react with one another. The nitrate ions oxidize the diphenylamine, and the chemical species produced is a blue-colored compound. Okay? So, we're good with nitrification. Okay. Now, third or second to the last part of the discussion is the process of denitrification. So, denitrification, we've discussed in the lecture, is the process that converts nitrate to nitrogen gas. No, we expect that yung nitrogen that is in the form of nitrite, no, nasa siya, nitrate rather. Na kasi pag nasa nitrate form ang nitrogen mo, bioavailable siya. It can be easily assimilated by living organisms, diba? So, sa denitrification, we're returning nitrogen to the atmosphere. Okay? So, from nitrate to nitrogen gas. So, denitrification processes that, or it's a process, rather, denitrification is the process of converting bioavailable nitrogen to nitrogen gas, to its gaseous or atmospheric form. Okay? So you expect the output is of course dinitrogen gas, no? N2. So balik na siya ulit sa atmosphere. Now for the processes or for the protocol, let's take a look. We have one tube of nitrate broth with Durham. We will inoculate it with 0.1 gram of soil and another with 0.1 ml of Pseudomonas aeruginosa. And then we fit the tubes with the rubber stopper. We make sure that no gas enters. So kailangan gawin nating anaerobic yung setup. And then incubate and examine for a few parameters: pH, ammonia, nitrates, and then gas. So gas, of course, you expect is nitrogen gas. Let's take a look at the data. Okay, so part 3, denitrification. So the pH of both setups relative to the control is of course more basic, pero in reality it might vary a little more, no? Hindi siya don't expect that it's going to be basic all the time. Pwede rin siyang maglaro sa neutral pH, no? But as time passes by, you expect that the pH of the samples during or the pH of a matrix where denitrification is occurring, you expect it to be more basic, less acidic. Okay. Now, the presence of ammonia could be positive or negative, right? Or from positive eventually going negative. So from day one of week or the first day of the week going towards the last day, pakonti ng pakonti yung ammonia na madedetect. While for Pseudomonas, also the same thing. Um, for nitrites and nitrates, also the same thing. No? Initially, you can, in the early parts of the week of observation, you expect to see them. But eventually, they will be negative, no? Or they will be gone. Now, for gas production, we expect that the soil sample or the setup with soil has more gas than Pseudomonas setup. Kasi, again, multiple organisms are present or multiple species of microbes of denitrifying bacteria are present in the soil. So you expect more gas to be produced compared to Pseudomonas alone. No? Okay? Remember, denitrification is carried out in completely anaerobic conditions. Okay? So, very important. Kaya nga important yung pagtatakip ng tube, no? To make sure that no gas has entered it. Tapos, talaga siya, completely anaerobic talaga siya. Okay? Then last part of our experiment is nitrogen fixation. So here, you're just asked to isolate and observe the organisms that are capable of symbiotic or biological nitrogen fixation, and observe also organisms that are capable of free or non-symbiotic nitrogen fixation. So nitrogen fixation, as we've discussed in our lecture, is a process where atmospheric nitrogen is converted to a form such as ammonia, no, or para readily ma-assimilate siya, or readily can be assimilated or processed or further oxidized to more easily assimilated forms, so like nitrites and nitrates. Okay, a few examples of microbial groups, bacterial groups that are shown to be or known to be symbiotic bacteria capable of nitrogen fixation. We have Rhizobium. We also have Anabina. Though Anabina is actually ang Anabina ay cyanobacteria, 'di ba? So capable siya of symbiotic tsaka free living na nitrogen fixation. And then we also have Bradyrhizobium and then Frankia. Frankia, if I'm not mistaken, mayroon din yata siyang may certain species that do not require nodule formation. Okay, so varied ang answer nyo dito. Hindi ako magpo-provide ng any additional information, but you just provide as ask, and then I'll just check on the plausibility of your answers. And then lastly, growth of microorganisms in nitrogen-free culture media. So ito naman yung ating free living. No? As one example of our free living bacteria capable of carrying out nitrogen fixation is Azotobacter. No, that's one example. It's a gram-negative microbe. And then we can, of course, just provide the description of the cells. And then if we take a look at the colonies, ganun din, no? May rin yan, for sure. We expect actually specifically for the case of Azotobacter, pigmented ang colonies ng Azotobacter, no? So you can provide additional description for whatever microbe, whatever bacterial species you chose for the nitrogen fixation part. Okay? So that ends our post-lab notes for module number 6. I won't be answering the guide questions, no? Kasi part na to ng exam, or some of them probably I will adapt to the examination, to the summative assessment number 4. No? Okay? So I'll make this announcement in this recorded session. I won't be providing you the I won't be activating the summative assessment this Friday. I will give you enough time to read or to listen to the recorded session, itong first part. And then there's another one that I will also post on ano naman, no, on microbial community profiling. So, Friday, dedicated time to review on the recorded sessions. And then I will activate the enabling assessment this Saturday, and I will give you 3 days to complete it. Because may part doon na magko-compute kayo gamit yung ano no, yung mag-implement kayo ng pipeline, yung Mothor pipeline natin sa Galaxy. I'll give you another set of sample data, and then there are associated questions. That's why I'm giving you 72 hours to complete that exam because it will take time for your machines to compute. Diba? Okay, so that ends our first recorded session. Then I will make sure that after this, proceed na kayo agad dun sa ating second session. Okay?